Good evening, everyone. Before I get started, please let me know in the chat that you can hear me correctly. So quickly put it down in the comments that you can hear me quickly. All right, that's great. Hello, one and all. Welcome. My name is Dilukshi, and I will be your host this evening. I know you all will be wondering, where is Shekhar sir? Well, since this is a 6-session webinar series, he will definitely be joining us in the future sessions. We just decided to switch it up in between. Thank you for joining us on day 3 of the 3D Bioprinting webinar series powered by Altem Technologies and Biotechnica. Before we begin with today's session, I would first like to give a heartfelt thanks to all of you dear attendees. Without you, this 6-series webinar would never be possible. Trust me when I say that all of your support, comments, and feedback goes a really long way. It encourages us and companies like Altem to bring you more such wonderful webinars. It's not an easy job to coordinate these events, but your support, that is what motivates us. Before we begin, I would like to share a quote which I feel is apt for today. It is by Brian Herbert. It says, "The capacity to learn is a gift. The ability to learn is a skill. The willingness to learn is a choice." So thank you. Thank you, all of you, for making the choice to join us here and being eager to learn. A small request to make this experience encouraging for all of us here at Altem and Biotechnica is to kindly share this webinar link with your friends, peers, colleagues. And also don't forget to tag us on our LinkedIn pages for both Altem and Biotechnica. Sharing is caring. I hope you all have shared the webinar link. A couple of instructions to note. There will be a Q&A session, so kindly post your questions with the letter Q and your question. So I repeat, there will be a Q&A session, so put in the letter Q followed by your question. There will also be feedback forms provided at the end. So kindly wait for our team to post the link in the chat. So now, without further ado, I would like to invite our speaker, Dr. Divya Shanmugarajan, with us to start today's session. Good evening, ma'am. How are you?
Yeah, fine, Dilakshi. Hope you are doing good.
Yes, ma'am. All good, and all of us are eager to learn. So over to you, ma'am.
It's my great pleasure with you all on the virtual platform and to express our version of what is a forthcoming technology in a cell culturing because we know about it. Cell culturing is going to be have an huge and massive impact in science and technology or the people who is doing working on a molecular pathogenesis and so on. So ex Dilakshi, it's a great pleasure to be with you all today, and it's my I welcome all from YouTube listeners, Biotechnica watchers, and Altem watchers for coming to this wonderful session. Can I start, Dilakshi? Is my voice is audible and my screen is visible to everyone? Hi, Dilakshi?
Yes, ma'am. You can start. You can start. Yes.
Yes, thank you so much, Dilakshi. So very good evening to all. It's my great pleasure to be with you all today from Altem Technologies and then Biotechnica to express more about the 3D cell culturing technology in vitro studies using bio 3D printing. This seems to be very interesting for you all because usually we used to culture our cells in 2D cell culture plates and then look for its metabolism and whatever studies that you want to perform. But this is some kind of a revolution where we are trying to transit from a 2D cell culturing technology to the 3D cell culturing technology to have a better improvement of disease models and to study in the better molecular pathogenesis, network analysis, and as well as to mimic exactly the prototype of in vivo. My session is going to be like this. You are going to learn about why we need this technology and why, especially with bioprinting, why I was keep on stressing the bioprinting with the 3D cell culture because we already been trying to move from a 4G technology to 5G technology, and the transition is happening in all over the world from one technology to other technology. And of course, in a science like a life sciences, we really need a great transition for a better improvement. As we all know that we are in a pandemic situation, we got a vaccine in a shorter period of time by utilizing in a computation chemistry, biology, and as well as an advancement in life sciences and technology. So like that, there is a one such a topic I want to share with you all today how this bioprinting with 3D cell culturing technology is really going to benefit the researchers, professors, and then enthusiastic research scholars. As I keep walking along with you, you can just come to know about interesting things that has been discovered and keep on moving in a 3D cell culture technology. So before starting the session, I would like to see that what is cell culture? Can you all put it in the chat? Why we need to do cell culture? Why I'm stressing cell culture, and why there is a need of cell culture? If you know the answers, please put it in the chat. What is a cell culture? I hope everyone started typing your answers. Before that, I would like to introduce my company. Altem Technologies is one of the leading company who has a different wings in various science and technology. One of the major impact or imprint in science and technology is with the BioVia Discovery Studio and the Cell Link, and where we have a solutions for 3DS technology. BioVia is made for life science and simulations, and the Cell Link is for 3D printing technology. And you can see our esteemed awards for various years has been displayed on the left hand side. And this is a company had a 3D innovation platform for all the solutions. So okay, now I would like to say about it. See, come on together. Today's challenge, and we are seeing this challenge all over the world. It is not only in the country, across the world we are trying to see these challenges. And then if for instance we know about it, certain medicines will be good for one particular group of people, like I could say that European people will have this kind of different genetic pattern, and Asian peoples will try to have a different genetic pattern, and other group of countries will have like this, we have a different genetic patterns. And then if you want to design a drug specifically for certain group of peoples, and when you take your journey for this drug discovery and development process, will almost take approximately 16 years. And then how we should say, for instance, there is a person who is suffering from certain kind of an unknown disease or an any kind of a disease which is rarely occurred. If you want to design a drug and you want to succeed in it, how long it will take? See, for instance, before that the medicine is going to occur and coming into the market, the journey of that person is going to be a mortal, right? So how do we take this? First challenge. Second challenge, and we all know about it, human transplantation. 21 patients will die every day due to the lack of the organ transplants. And then this organ transplant, even though there is a transplantation occurs, there will be a lot of grafting issues will occur, and that need to be taken care. There will be a lot of things that need to be taken care immunologically when we want to transplant your organs. And they are not very, we don't have such a group of peoples to transplant also. And what we can do with, what is the alternative way of doing it? That's how we have to think about it. Is there is any way? Of course, we have a solutions for this. And then, as I said, in a pharmaceutical journey of discovering a drug, researchers are spending more than $2 billion, one more than I could say $3 billion, and then the rest of the $2 billion are going for a wrong testing methods. Why it's wrong testing methods? We have been doing compound explorations and then taking the compounds and then doing the assay and then doing the assay. Okay, I mentioned this, but doing assay, performing, and then transforming to the in vivo. There is a gap is happening. What we are doing, the method testing within a monolayer cell culturing technology actually. But what is our system is a 3D, right? Everywhere all cells are be communicating each other, there will be a communication and there will be a message will be sent. As soon as some person pitches me, I will just like that, I will, there is a immediate response will happen in my system. Why it is happening? It is sending a synapse to my brain and telling that there is something is happening. So it is a 3D system, cells are keep on communicating each other, but when we study in a 2D cell culturing, there is a drawback. How do we address this? So today is a great challenge in the medicine can be a solution through a bio 3D printing technology. I can say that very surely. So what are the things that we can study in bio 3D printing technology? Let's make it so interesting about it. Say for instance, this is a system. Or you enthusiastic researchers want to study the tissue growth, and if you know about a tissue growth, how the tissue is growing, it will be very interesting to know about it. And then cell behavior, what are the cells behavior is there, how the network is forming, how the messages keep on sending, whether what is the intermediate product is separating. Would you like to know about it? And simulation of the cells, live simulation, how the cell is really inside the body will keep moving. Would you like to know about that? And 3D frame of your living organisms. You want to create the 3D exact in vivo frame of your living organism. Signaling, drug metabolism, very important for pharmaceutical industry who want to study the PKE and PKD studies, how the metabolism of the drugs will happen to the admit properties. And you want to reduce really the rate of your drug discovery process, and want to success your new compound entities in a clinical trials, and want to save your $2 million. What is the solution? Bio 3D printing with the 3D culture technology is solutions for all these answers. Why I keep on stressing this bio 3D printing, bio 3D cell culture? We have a solutions. There are lots of people who worked on and gave the solutions to prove that 3D printing with the cells could be an exactly 3D in vivo system for your testing. So when we use this technology in an efficient way, that will solve all these issues. Let's keep moving along, walk along with me and know how this really I'm going to show you even the drug metabolism, how the simulation of cells we are going to see the how the heart will be beating will be there when the cells are trying to grow, how the signaling is going to move on, how the tissue growth is going to happen, what are the enzymes that is going to secrete when the tissue is growth is there. We have a solutions, and as keep working on, you will know about that. So before that, we want to know about a 2D cell culture. What is really the disadvantage or the demerits in a 2D environment? You can very well see this circuit organization, right? This is how the circuit organization will be in vitro technology. Okay? And then you can see the cells are being communicating, and there will be a synapse will be keep sending, and this is the environment of the in vitro exactly. Okay? And this is, doesn't looks like a real cell environment, and there's a little bit lack of productivity. How do we say this is lack of productivity? As you come to know about it very soon about it. And there's a lack of productivity between the cells, and one period of time what will happen, there will be a issue will be in a growth media, and there will be expansion of cells only within this particular plate we are going. And once the new tissues are going to deplete, that will trying to damage your cell environment. And in that situations, in that position, you are going to test your drugs or in a compound. It could be any compound, organic, inorganic, or maybe a natural compounds, whatever is it. You want to discover something into it. Especially, I could even say that you want to develop the SARS-CoV-2 model, and you want to really study whether this particular natural product or then a drug is going to work it out. We cannot work it directly into the humans, right? So that how we can develop even disease models using 3D cell culture and 3D printing technology. And then if you're going without that, when you want to execute this process, that will increase the cost, failure rate of your new compound entities in your clinical trials. And as I say that this is really this is not really a biological model, and there is no cells to cell communication or cell to ECM extracellular matrix communication. So how do we shift from the cellular communication? So we are going to be take a shift, and we are going to enter into an environment called 3D. This environment is going to be more relevant. See the structure and see how the communications are happening. That is in a circuit organization in 2D. This is a circuit organization in 3D. There are lots of cells you can see, that lot of cell types you can see, that is keep on communicating, and there is a signaling will be keep on moving, and then a better simulation of this living organism. So when I keep speaking right now, there is a lots of metabolism is happening and keep sending me signals to my brain to my organ, and to my, that's why I keep moving my hands. This is a synapse happening. It's not a one such a cell is happening. Lots of cells are keep on communicating each other. But when you do it in 2D, it is not exactly the relevant model, right? So better simulation of the living conditions can be achieved only in 3D environment. That is a realistic environment that we are trying to mimic and going to give it. And when we try to use this, and we can, of course, we can reduce the animal models where you want to test your animals with the different groups, and you want to study better in the future without introducing animals. We can do the cell culturing with 3D printing technology, reduce your animal models. So it could be very happy. Animals will be happy in future. Okay, and then you want to reduce. You want to create any kind of disease models, and I can see that in when Aishwarya was trying to give a session, people was keep on asking about the disease models, right? Disease models is feasibly can be done by using this technology, actually, and we have the proof for that also. As I told, I keep moving on. You will see the solutions for them, and there will be exact cell to cell communication and cell to ECM signaling will be there, and this is exactly the prototype of an in vivo system. So 3D environment is going to be exact prototype of an in vivo environment by printing your cells with 3D printers. So that's a shift we are going to take to have to study the models and to mimic exactly the in vivo systems. So pharmaceuticals. So there is a lots of I could say that when who is doing working in the pharmaceutical industries, and they really want to do an exact studies, and you want to reduce, and you want to go for clinical trials, this is the best solution, I could say. How they can improve the pharmaceutical workflows. Compound exploration. I have millions of compounds, and I would like to know which compound is the best. How do I do that? I have a separate solutions for that. That is under BioVia, we have a separate solutions. I come to know about it. There are few molecules, or I could, and from a millions, I have drawn within a few hundreds. So I have compounds with me, and I want to do the characterization. How do we do characterization? Assay development. So far, they are using only the 2D cell culture technology. And once they do the 2D cell culture technology, they are moving into preclinical testing with the different animal models, then moving for a human clinical trials. Here, exactly the failure rate will be more. So to replace this, we are introducing a 3D biofabrication technology that will reduce your cost and reduce your money and increase the efficiency. So for a better improvement of pharmaceutical workflows, and to have to reduce the false positive rates, and to increase the drug efficacy and biologically efficient molecules, this is a way where we can improve our workflows. Okay, so this is one of the example of pharmaceutical workflows. I was keep on stressing why bioprinting with the cells. Why bioprinting with the cells? Say, when you want to print your cells, to mimic exactly like the in vivo system, there should be certain instrumentation, analytical instrumentation should play a role. That's why the 3D printing bioprinters are playing a major role. With automations and going to use a smart material, that is very important. What is the smart material? The smart materials are your bioinks. What is this bioink actually? Why I'm telling this biology, is this biologically made ink? Of course, this is a name actually. But For an system, say, for liver cells to grow, there's a different environment. For the heart cells to grow, there's a different environment. And you want to grow your skin tissue, there's a different environment. So the smart material that you decide, that also must be much more smarter, right? So you have to bring a very smarter environment for your cells to grow. That can be done by through bio inks. So this bio inks, how do we really get this bio inks? How do you get it? So we have a solutions for that. I will tell you in the next few slides. We are going to have a smart material that is called a bio ink that will help you to print your cells using bio 3D printers and a 3D cell culturing for or a 3D printing for an organ chip, whatever is it. And then that bioprinted structure will exactly looks like a native structure. You can see the structure of a 3D printed construct that is engineering to a myocardium, and that is exactly looks like we have a blood vessels, we have a myocytes, and then the communication between the cells, and looks exactly like a layer by layer. But in 2D, we have only one layer, but 3D, we have several layers. That's why we have lots of print heads in the BioX. So we cannot print only one cells. We need multiple cells to be, multiple layers need to be printed with a different, whether with a one print head with the one cells or a multiple print head with the multiple cells. That is feasibly can be done. So this will try to improve the cellular behavior and gives exactly a native-like structure. And moreover, there are lots of problems will occur, try to occur that will be potentially resolved or elevated when we want to use this kinds of technology, or when you want to test it in your 3D printer construct. So what are the advantages? Definitely, as the technologies keep moving on. See, initially when we started initially writing with an normal pencils or like that. Now we are having an different kinds of pens to write it in calligraphic. So like that, right? We are taking a different shifts. So similarly, when you want to give a change, there should be a transfer should happen to increase the efficiency in the drug discovery process. And this is a cost-effective way of in a pharmaceutical, as is for researchers who is working on cell biology, molecular biology, or you want to really study the drug metabolism and pharmaceutical industries and pharmaceutical molecules and so on. And this will save your time and reduce your preclinical testings in animals, and this where you get a reliable outcomes. So that's why bioprinting with the cells, with using for 3D cell culturing technology, or give you all these benefits for you. So that's why a transition should happen. So when you go for 2D, what will happen? The molecular mechanism will be associated with the diseases. And you want to study, there will be lots of drugs will be failing, and it's a poor efficacy. This is how the 2D cell culture model will be being. And when there's a transition is going to happen, there's a success model and a human diseases. Disease modeling is the most important thing. And if you want to really study how exactly the disease model should be built, and you want to recalculate exactly the human microenvironment, and you want to study on cell behavior, and you want to study how the cell spread is happening, morphology, polarity, gene expression, protein expression, viability, mortality, proliferation, different. I can keep on telling all these words because all these things are being connected to the converged together only to an, cell cultures. And you want to do all these things and to represent and simulate the cellular environment, so that can be done to an 3 cell culturings. And this will significantly accelerate your drug discovery process and prevent your late clinical trial phases. And I was keep on telling there isn't anything which is already performed that will keep cracking our brain. Yes, of course, there are already succeeded things which happened through this cell culturing technology, which I'm going to share with you all. 3D revolution. So when you want to embed cells and incubate and analyze, and that we are doing in a culture tissues and growing and giving a 2D shield that will try to have an poor cellular environment. Okay, and negative influence from the substrate. But whereas then I'm going to have and I say very simple, I wanted to study in some cancer models, adenocarcinoma models. I have the cells. Either you can take it from a patient specific personalized medicine. You're always welcome. So if you for personalized medicines, what are now many countries, Western countries of the performing right now, and they keep on testing. It's a personalized medicine. You want to do that? Of course, we have solutions for that. Take a patient-specific cells also, and then do this primary initial steps is same similar in both 2D and 3D, but only when we come to the printing that will be different and incubating will be different. That's it. And we are going to culture our cells and incubate it and bioprint it and into the and get a 3D model that is going to have an you can see there is a green color that is physiologically more relevant model. So 3D revolution must happen in a cell culturing technology using 3D printers to have a good intercellular communication between the cells. Materials. So I told her this is a person who is a hero actually. Okay. So material is needed for success. So selecting your wide range of materials, it should be standardized by your researchers. Which environment you want to mimic exactly, and say for instance you want to develop a cancer model or liver tissue cancer model or a heart tissue cancer model, whatever the model you want to build, that the bio inks are very important. And then without the succession of bio ink, whatever you are trying to print, that all is going to be a little bit or going to lack, I could say that, because this is a one which will help you to understand or help you to go for a further development all your cells. So material is very important. So from a cell Altem to cell link, we are supplying about various raw materials also, concentrated compounds, stock solutions, very basic bio inks. First, you want to try with the basic bio inks or ready to usage products. That means it's instantly available. I want to print an heart cells, let me say in industry exam, heart cells I want to build. So there is a special ink for that, a laminate, a 521, there is an ink will be there. I'm going to purchase that ink, and I already have the IPSC cells, which is derived from a cartilage, it's ready for me. I'm going to mix these two things and I'm going to culture it, I'm going to print it with 3D bioprinter. I'm going to have an in vivo environment. So testing will be easier. So standard we have an bio inks you can purchase directly, or in case if you want to develop really the biometal, of course we have to concentrate on various parameters like biophysical parameters and the biomechanical parameters, because rheology is important, how much consistency is very important. All these things can be standardized and you can use it also. But this bioinks or ready-made, which is already been standardized and it is ready to use for a person who want to do a fast research things. And there is also advanced bioinks for specific materials, and each bioinks has its own properties and cross-linking technology. And tissue engineering kits is also available. You can buy a kit. How we do in the molecular biology, we buy a kit for an DNA extraction and then RNA extraction, and you want to do for a uh, um, capillaries, all these things. Tissue engineering kits are available, like how the molecular biological kits are available. Tissue engineering kits are available where you can use it and we can use it. So use in your bioinks, it's very important for your advanced research cultural technology. So materials is a hero, and we have to be more concentrate on the materials to be get success in your bio 3D printing. So how do we do this? So it's a very important protocol. So you'll be thinking about it. See, I was keep on chilling 3D cell culturing. How do we exactly do that? It's very, very simple process, I could say that. So grow your cells, embedded cells with a bioink, and then put it in a bioX or a bioprinter that will print and give you the cells. So growing cells, embed with a bioink and then print it. Any type of a cells can be printed along with a specific bioinks, and then you can also demonstrate and you can see the various cells that have been printed and that is succeeded by on a cell link and Altem. It's by means of cancer cells they have printed, liver cells they have printed, plasma cells they have printed, stem cells they have printed. So like this is there is no limited with the cells. What cells you have to be very specific, what bioink specifically you have to be very specific and print with the bio 3D printing model. This is how it will go. So now we see how exactly what is the difference between 2D and 3D culture, and then is it really giving you the 3D environment or not? You will be keep thinking about it, right? I was telling what is a 2D and 3D. If you see the confocal images of these things, then I think it will be clear. You can get a more information about it. 2D versus 3D. Always a square cannot be cube, right? I think you all agree with me. The square is a square, and then it has only four sides. And the cube is always a cube, which try to have a more edges and sides and corners. That we have lots of things can be done, and very exactly take a 3D environment. Similarly, it is applicable in a 2D versus 3D cell culture technology also. So let's see here. I'm going to take you three different studies, and then I'm going to share you the confocal images. First, I'm going to show you HUVEC cell line, which is co-cultured the fibroblast. Okay. This is a 3D environment. It's required for development of complex structures and network formations. Very important network formation of HUVC cells. And then you can see that 2D cell culture in a day 3 and the 3D cell culture in day 14. So why I differentiated the dates? Why I should not take a day 3 and I should show you? I'll tell you the reason. So first see that, and this is in ZO1, one is red in color, and GFP is a green color, and nuclear staining with the DAFI is a blue in color. Okay? And then on the other hand, when you see this, and HUVCs, okay, it is co-cultured within a fibroblast, and then with the gelba fibroneck is one of the special bioink had been utilized, and then you can see that network formation, that is a one which is I was trying to stress over here, network formation. The network formation, you can see it in a day 14 of 3D cell culturing. It's like an exactly mimics like your in vivo systems. But you cannot see that network in a 2D cell culturing in the day 3. Moreover, when we try to use a 3D cell culture, long-term studies of your drugs or any metabolism or expression can be understandable, can be noted down. And but in 2D cell culture, due to lack of an environment or in a substrates and so on, and they not mimic like a micro environment like in vivo, we cannot study for a longer term studies. This 3D cell culturing and network formation with an Chelma bioink will give you a 3D to cell matrix signaling, and then the 3D support we can be aided for developing a more complex vascularization model. Is very important. You want to develop the complex vascularization model because our system is like that, right? It is a very complex model, and there will be lots of signaling will be coming. And you can, when you think about a metabolic pathway, just look about the metabolic pathway. And if you are already working network pharmacology, think about that how these interactions will happen from one cell to other cell. We think that PPR gamma is playing a major role in especially in your I mean insulin signal, but that is also one of the protein which is playing a role in cancer also. And one target could be have a roles in a multiple disease models. If you want to study exactly that, we need a time, we need a network, we need a exact days need to be done. That can be done only with the 3D cell culturing technology, long-term study of your cells and behavior, sprouting, networking. And again, I just want to show you another one example. If you are a cancer researcher, I think that is a very big benefit for you all. Lung cancer markers. As soon as how do we say there is a cancer has been exposed, the person is having a cancer, we will be just trying to detect the markers. Markers are very important. Right? So lung cancer will have a marker 3D cell culture, exactly looks like the in vivo expression. And there's a specific marker called beta catenin, shows more relevant like a 3D model in A54 cell lung carcinoma cells. And you see here, beta catenin. Beta catenin is a very important, and there is a cell-to-cell communication, and there will be a cluster around the nucleus. And you see this is there to be a contact and aggregation clusters around will be there, but you cannot see that same cluster in your 2D cell culturing of the day 4. And then how we say this in a day 14, but as the cells keeps on growing each and every day, we'll be keep on monitoring its morphology and cellular behavior and how exactly cells are looking like. So in the day 14, exactly there we can see this one beta carotene expression. And then you can also see that there is exactly a more physiological relevant model, and you can see the exactly how the cell communications are happening. Then we try to use these kinds of in vivo resemblance like expression like model that will improve drastically your therapeutic application and reduce the time to your market for any drugs. So ultimately, it will reduce your, say, if you are going to spend about 16 years for this kind of a study for drug discovery process, almost 2 to 3 folds it will reduce your time. And you are going to your interest, you are a person in industries who is investing $2 billion, $3 billion. So then we use this kinds of a smart technology, 3D cell culture, 3D printing technology will reduce your 5 folds of your money, saves your time, increases efficiency, better output. That's what we'll be seeing in 3D cell culturing technology. Again, that another example, and you can see that arrangement of liver cells, how the organotypic and phenotypic behaviors are revealed. And we know that alpha smooth muscle actin on the day 4, and here we used a call one. It's in a bio ink culture the same tissue and HPG2 to see how the HPG2 arrangements are there in a 3D environment, and it is a green staining and showing that exactly like the in vivo. So alpha smooth muscle actin will try to reproduce and evaluate. See, it is a valuable tool. See, if I say that is a valuable tool to evaluate the process, it's a valid. This is one example. Alpha smooth muscle actin evaluate your formation and localization subcellular compartment of the cells. You can see clearly the arrangements of the cells that shows organotypic and phenotypic behavior. And this is a border which you're seeing in 3D, which is not seen in a 2D. That shows and confirms that. So even phenotypic organotype behavior rearrangement also, this seems to be perfect. Okay, perfect. So far we have seen various examples. Now, if I show you the protocol, how to print the cells, and also I will say the gene expressions, because day by when you keep studying about the cells, the expression of the cells will vary from a day 1 till we study for the day 10, something like that. So here an example is a 3D printed using a BioX and then of a biopod laden using cell construct for in vitro evaluation to see whether exactly it is like in vivo or not. Let's see this example. So bioprinting, it's very easy. This is a protocol. Okay. So if you want to do printing in future, simple protocol. So here this is I'm going to print a cornocyte laden cell link construct like to evaluate for an in vitro studies. I need a blueprint for this. What is this blueprint? The structure which you want to print, we call that as blueprint. Which structure you want to print, and then what should be a dimension? How what should be a layer? What should be a thickness? Everything you have to decide for yourself. How do we get this blueprint? You can use any kind of smart CAD tools. Okay, we can bring any CAD tools and then bring the structure and take that blueprint in a .stl format, and then you can, when you have it in a pen drive, of course we have an option to insert your pen drive or USB in a BioX, there you can insert, and even the same technology you can use it with the Luminex also. If you want to build in a more vast structure, that basically uses a DLP technology. BioX basically uses extrusion based printing. Okay? I have a cell link cartilage, it's ready. When I want to load, I have a nasal cornocytes, and then I have chosen a superhero bio ink. Okay, he is a superhero because without him or without her, we cannot print any of the things. And I all being mixed and kept it, and I put it in a Lana Afro, and I'm trying to print it. And then for going forward with the cross linking, this cross linking will vary for an cells to cells, and I could say the bioinks to bioinks, it will vary actually. So some will have an a UV, some will have an a calcium, some will try to have a different kinds of caustic agent. That is not an issue. When you get a bioink and you want to study, you can specifically use that kind of an analysis. And finally, after 4 weeks, we get an air construct. So this is the one example to for an in vitro evaluation. How do we say? How do we evaluate it? I printed it. How do we evaluate? There should be an secretions, and you can see that cardiocytes will try to secrete glycosaminoglycans. GAG is very important to function to properly. It is second in a chain to function properly. It is a physiologically controlled and controls the maturation speed. Okay? And you can see here in the day 14, there is a limited amount of GAGs, only 22 or 21 UG. But in day 28, so how long? Day 28. When we use this today, what will happen? By the time the cells might have been depleted or died, or it could be gone for some other state of your cell cycle or in a cell phase like that. Day 28, it's still surviving, and you can see the GAG expression. Okay, and also you can see the other components. So aggregate, also aggregate structure production will increase together with the collagen type 2 cell, which forms a major structural component of the cartilage. Along with that, you can see the myelin and compy. So that shows, and the cells are producing what they exactly want it in the body, how the in vivo, and this is a greater indication that this is a good model to study. This is a kind of an in vitro evaluation that they made to confirm that 3D printed with the bioprinting cells will have a more efficacy. Okay. I was keep on telling only the stories and all understanding. If you see something more interestingly animatedly, when it keeps simulating, then that will be very interesting, right? So developing a heart tissue model. This is another technology, and then this is a most famous technology. I could say that many people we are seeing cardiomyocytes will be a major issue, and we can when we can develop the heart tissue model and we can study the cell's behavior, then that will be a great solution. How do we do that? Being the simple answer, 3D printing, but the 3D cell culturing technology is a solution. 3D printing the cells. Let's take you for a little small video for you. Let's see this. So this is a BioX. Now we are going to print it with the BioX, with the cells. And this is a very simple protocol, not like a much too much of assay development, standardization. It's very simple. You can see this. Oh, sorry. Yes. So any instrument, any analytical instrument, what we'll do, we'll try to calibrate it, right? So same calibration we are going to do here. After doing calibration only, we are going to print our cells. So calibration is also auto calibration. We no need to do any manual things also. See. Usually the calibration they will try to do it with any test printers. Okay, any test inks will be there, bio inks will be there. We used to use that. Then only we'll move with the printing with the cells. And very important, whether you want to print it with a culture dish or an 96 well plates or whatever well plates you want to use, make sure you place that. Now they are doing calibration. Calibration need to be done for all the print heads that you are going to use. Okay, that's why they are checking it. and now they are giving a test print. Step 2, we are going to print with the cells. Let's see this. We are in a step 2, printing procedure. Now they are placing a culture dish. See, anytime you can pass the print at any time you want to left, how many layers you want to print, how much control, everything is in control, and everything you can able to do it by means of a codes. Okay, it's very simple technology. So this cross-linking will vary depending upon the bioink, as I stated earlier. Now they are using cross-linking agent, and they're removing the cross-linking agent. Now load your culture medium. That's it. And then finally incubate it. So happy printing with BioX. So this is how the 3D printers will help you to print your cells for a 3D environment. So now I'm telling about 3D model, right? Now see this example. This is one of the disease model I have chosen, especially cardiovascular disease. And I want to print, I want to make it some kind of a compound exploration. I want to bring some kind of new drugs to the market, especially to treat this disease. So bioprinted by the cardiac muscle tissues, and I want to mimic a disease condition. That is very important. So to develop this, I need an what I need basically I want when I want to print my cells for the 3D printers, I need an cells and I need an ink. So heart tissue model, which has been derived from an pluripotent stem cells (IPS) from a cardiac aggregates, and then 3D printing with the cell link lamin ink 5:1 bioink, especially for develop your heart tissue. So we are going to see how the culture is going to be for the 3 weeks. Day 1 up to 3 weeks, what are the changes are going to occur and what are the functions going to happen. That's what we are going to monitor. Even you can see the beating of your cells constructs. So now see the day week 1. So when you want to print your cells, simple. I have got the model, and that is like that is a STL file I was telling about it, which model I want to construct. I bought the model and I filled it, and then I'm printing with the cells. So derived iPSC human derived cardiac cells, lamin ink 5:1 bioprinting. So this model can use to utilize for an to investigate cardiomyocyte maturation, drug screening, identifying a drug targets, and ask this for your cardiac regeneration. How do we say this? I'm not going to say, I'm going to show you. Beating of cells at the day 7. See, when you want to see how exactly the cells are going to beat, and especially the cardiac tissue, in 1 week of culture. See the aggregates integrated with the bioink. We use a special bioinks, right? Let's see this. How these aggregations are happening. It is integrated with the bioink. And you can see the stretching out and pulling of this cardiomyocyte sheet within the bioink. which is very important. That's why you say he's a hero. Bioinks are heroes. And this bio ink will help you for cell proliferation, reorganization, attachment, functionality, and metabolisms. So within a week of cells, you can see this development as an indication of your in vivo systems. Beating of cells at day 7. Now, after 3 weeks, now cell has grown well, and there should be a flux will happen, right? There will be a flux will happen inside the systems, and we have to see is exactly the contractions are happening or not. Let's see this. And there's the aggregation of correlation of flux will happen during a contraction, and this will be an intracellular immobilization of calcium in your live cardiac aggregates will happen, and you can see the synchronization and green flashes that indicates the cell to cell interaction and communication of the tissue model. That shows this is a disease model that we developed to study the cardiomyocyte functionalities. So this is the overall representation. So bioprinted art tissue to understand the cardiovascular disease and advancement for any kind of therapeutic discoveries or new model discoveries. The day 1 and from the day 21, week by week, you can able to study its behavior, and then you can see how exactly the contractions are happening. So creating an exact in vivo disease models with 3D printing, with 3D printer, 3D cell culturing, and 3D printing technology will benefit for better therapeutic applications. So how do we do real-time environment, which is most important? Take it from patient. How, for example, how the medical imaging will happen. I was trying to compare over here. Medical imaging, model of defect, and they will try to have a 3D printing template. And they go for 3D bioprinting. Here it's simple. Take it from patient. That's why I was telling you. Personalized medicines. That is also feasible here. And take it cells from the person and use a bio ink as a polymer base and introduce what are the growth factors that you want to test and you want to see for the drugs. Print the tissue. Happy printing with BioX and with the 3D cell culture will give you a solution for all your problems. And what are significance? So this is going to be a future regenerative medicine. And you say, for instance, there is also, if you want to print a layer by layer of cells and you want to have more layers, especially with the skin tissues, you try to have more layers of cells you want to print it. Of course, you can see that we can load with the, I showed you in a BioX with 3 different print heads, right? So where we can load along with your bio ink and cells where you can have a 3-dimensional direction and also the layer by layer, that is most important. And incubate it and print within and you can go for testing. And this really generates a similar like your real tissue, and where we have a good high cell viability to display the great printability. All the things which I showed you in the previous slides that shows the cell viability, how long the cells are viable, and with the display the great printability within the BioX and various 3 bioprinters. So you'll be now inspired, right? Of course, I believe that you people now inspired. So then you want to spend your time on the such application. They already done and with the different cells, with the different metabolism. Drug uptake and metabolism in the intestine using the specific bioinks and CO2 cells in your intestine and liver and in the pancreas. And of course, the cancer models is also done. Skins and cancer tissue model also developed using 3D bioprinting technology for layer by layers and investigate a spheroid formation for lung cancer cells. What else is left? Definitely the people who is a neurologist or people who is interested in studying a neurological function. Yes, here's the solutions for you all too. Nervous system. To study the communicating and electrostatic potential, they are going to use basically they use a GELMA bioink, and then they study it along with the carbon nanotube enhancement connectivity and neural tissue and spinal cord neuroscience, how the motor neurons and astrocytes are being communicating. That's also they study. So this is actually it is since I have a lack of time, I'm trying to show you only with the limited number of slides, and then the achievements and inspirations happen with the 3D cell culture and 3D printing technology. Follow us in Altem and also in Cell Link. You can see lots of things that we keep on publishing day by day. Our products, Incredible Plus, BioX6. You want to do more than 3 layers, of course, we have solutions for that also, BioX6. And you can very can 6 layers of 3D printed matters. Of course, that is a huge thing that we can do it with the BioX6. And we have for an BioX MDX, BioX MDX Plus, Luminix, where we can have where we want to print exactly like this in a more complex vascularization structures. Using an extrusion-based printers or light-based printers, you have a solutions for your 3D printers. 3D printers have solutions for 3D cell cultures. Say for instance, we are also being into an in silico technology, computation chemistry and biology. We have solutions for all the things, right from a target discovery, sequence analysis, modeling, simulation, designing a library, pharmacokinetic and dynamic properties. Before coming into this 3D cell culturing, we try to screen these molecules with this using our BioVR Discovery Studio software. One of the solutions for computation biology and chemistry where one suit has a different protocols and solutions for all your problems. And they can do optimization screening. And once you finish, come to 3D printers, bio 3D printers, USL 3D cell culturing, all kinds of graphic technology. And this is a small video for you all to show about how Discovery Studio is going to work it out, especially in simulations. You want to see the when you try to give an external pressure or a temperature, definitely there will be an changes will happen in secondary structure analysis that can be seen lively when you want to see the simulation discovery studio. And you can see when I was trying to use this model and when I try to, after doing simulation, you can see secondary transitions happening, especially in helix, it's turning into the coils and with the different conformations at the different time. So it is not only limited, it is having a tools for pharmacophores, receptor ligand interaction, docking, and then simulations, X-ray crystallography. It is not at all limited with any tools. We have, and that's I told, it's a one suit for many solutions. And you want to do the constraint dynamics, especially within the region of binding region, you want to see the interactions. Of course, you can see even that also fix it with an active site amino acid to see whether the interactions are what happening over a 50 nanosecond or 100 nanosecond. We can see even that using our simulation tools, which basically uses a charm-based simulation for speeds and various models of editing, scening, and then various representation, better representation of your structures. All things we can able to see by using a driver your discovery studio softwares. So what are the other capabilities? If you are interested, you can see that Discovery Studio has a wide range of capabilities, having more simulation tools and then designing antibodies, engineering your protein, and you want to analyze your protein, predicting a motive, developability. We have solutions for all the things, right from macromolecules to small molecules. Macromolecules, simulations, and screening of your drugs, small molecules, scaffold coping, fragment replacement, pharmacophore modeling, structure-based, ligand-based. So we have all the solutions in the one suit in a name called BioVR Discovery Studio. And you want to predict the animal models, especially we have models for animals also, rat, mice, and the daphnia. Environmental models is also available where you can predict the dosage rate that has been developed using machine learning algorithms. And this is an example. Not say for instance, I have a small number of molecules. Of course, I can go ahead with this. If I have a large number of molecules, how do I go by my means of this one? And I want to automate it. I'm very sure that even I have a millions of compounds, I want to automate the process. If that is the case, we have to look it for a pipeline pilot where we can have multiple tools for multiple solutions that we want to, you are an interested scientist, you want to develop a new protocol of your own and you want to optimize the algorithm and want to enhance your research, then pipeline palette will give you all the solutions. And you are an aspirant programmer, you have a solutions even over here. But in terms of not only in terms of life sciences, we have imaging and we have tools for analytical instrumentations, machine learning, R scripting, and even a native layers for Java. Everything has been put together in one platform called Pipeline Palette. And that comes under the name called Dassault Systems Pipeline Palette and Dassault Systems BioVR Discovery Studio. And small molecules redesign, define your objective, do 2D simulation, 3D simulation, define your tool and pipeline your process, build your machine learning tools, and generate therapeutic design. This is also going to be very interactive and interpretive. and analyze your results using structured active relationship, registration, and finally come out with the compounds. So finally, so we want to screen, identify, optimize, and want to bio cells, bio ink, 3D printing. We have a solutions in Altem technologies, right from an in silico to an in vitro to mimic like in vivo systems. We have a solutions from a bio VR to bio 3D printers for drug discovery and development. And where and all you can use small molecules for agrochemical screenings and biotherapeutics, antibody designing, enzyme designing for detergents, food industries to develop the flavors, biofuels for ethanol, biodefense. So we have applied solutions in all these areas already. So finally, thank you all. Follow us on social media, Facebook, YouTube, Twitter, and here are the solutions for you all, and here are the questions for you all. I think it's the right time. I think it's almost 8 p.m. And what is a bioink? And the people, you can see there is a scan has been given over here. You can scan this QR code and answer your question 2. And then there is a conditions for this. This is the conditions. I will share you that. There's a contents tools are there. Say first of all, share your comments and feel free to share your comments and follow our page and try to tag minimum of your fewer friends. And the 3 winners will be selected randomly, and you are eligible to receive the awards. And the winners will be always will be announced in the next session. And all the questions need to be answered, and you'll be giving an Amazon gift voucher of rupees 2,000 rupees. So sitting at home and then seeing the things virtually and answering the small questions and gaining a knowledge is going to give you a worth of an Amazon gift voucher. Please utilize it and then follow us in all these pages, and we have solutions for all the life sciences. So one platform for various life science solutions: computational biology, chemistry, 3D printing, big data analytics using machine learning from Altem Technologies. Thank you.
Thank you, ma'am. That was a very, very informative session, and it went by so fast, and it was so interactive and so fun. I just love the way you presented it.
Thank you. Thank
Also,
you.
yeah, also, like ma'am mentioned, there is a contest that is going on. So you can
Yes.
scan the QR code. Please go and follow their page on LinkedIn as well. Additional
I,
rules will
I will
be
put that
mentioned there.
one. Yes.
And
Yes.
yeah, so they're fairly simple questions. I can see a lot of comments already giving away the answer, but please go to their page, follow the rules, and you all will stand a chance to win some nice prizes just for attending this
Yes.
session. Come on, it was a
Yes.
very,
Yes. Of course, you are going to get a worth of 2,000 rupees gift voucher from Amazon.
yeah. So
Just
for a free webinar you all attended, this is what you all are getting. So come on, guys. So do attend the subsequent webinars as well, because this is an ongoing contest. So it's at the end of the 6th session when you've participated in all that, randomly winners will be chosen. So please don't forget to join us in the next session as well. Okay, now we are kind of running short on time, so we really need to get the Q&A session going. So I'll choose a couple of questions. Don't worry, we'll have more sessions. 3 more sessions are still left. So that time we'll pick up your questions. So ma'am, are you ready to answer a few questions? We need
yes,
to...
yes, definitely, definitely. Please go.
Okay, fine. So we have a question by Sajari Roth. I'm sorry if I'm mispronouncing your name. Where exactly can we get the facility for 3D cell cultures? Ma'am.
Cell culture you have to buy. Okay. Then you have to print it with the bioprinters to get in a 3D cell culture.
Okay. So I hope that answered your question. Now, another question by Debjani, which says, how bioprinting plays an important role in the production of disease-free plants. There were many questions regarding, is it possible to do that with plant extracts and such?
Say for instance, first of all, for your query, I will say that develop the disease model. Say I have already given an example for a cancer model or in a cardiomyocyte model. Develop the model initially, and on the other hand, since you say plant extracts and so on, extract the compounds and then try to take the compounds what are present and test it for your bio 3D printing. I mean 3D cell culturing, and see the behavior. To understand about in vitro evaluation, I have so there will be a lot of biochemical reaction and enzyme will keep separating as the days goes on. So evaluate it and test it and see for its efficacy.
So, Devjani, I hope that answered your question. And the last one is by Swati Sharma. I'm working on skin cells and growing them in 3D Paul matrix. Ma'am, can we develop an artificial skin like growing together fibroblasts and keratinocytes and endothelial cells with this technique?
Yes, of course. If you want to develop a three different kinds of cells, choose I will say the solution. First, choose a which bioink you want to use, and then mix it with your cells that you want to print. You want to print it in a layers by layer, and you want to start it. Of course, you can do that.
Okay, Swati, I hope that answered your question. Really sorry, guys. Pretty short on time. There was a lot to take in, and I think ma'am answered most of your questions actually because this webinar, the presentation was so good. So that's why let's see next session what can be done. So, yes, we'll be wrapping up with this session. Apologies, but we need to respect the time limit. I know you all have a lot more questions. I can still see them coming, but we have 3 more sessions to go, so it can be picked up then. So I would like to say thank you so much, Divya ma'am. This was very highly and very informative. Like, I'm at a loss of words because even the questions are coming up, but most of it you were already answering in your PPT. So thank you so much, ma'am. It
Okay.
was such a fun session and it went by
Thank you.
so quickly. So honored to
Yeah.
have you here and such knowledgeable people. And thanks, a big thanks to Altem also. So everybody, like I said, encourage the speakers, support the speakers. It's because of your support and encouragement that when we feel like doing many more sessions like this. Just look at how
Definitely.
informative this has been, and it's a fun session as well. Like, you're getting to learn something new just sitting at home 1 hour, and at the end of it, you're even winning something. Possibilities are there. So thank you so much, everyone.
Thank you,
So
Dilipshi and Biotechnica for this and Atom Technology. Follow us. We are having very interesting sessions in future also. Keep following us. We'll be trying to deliver everything. I think so the knowledge is free for you all and you can learn a lot and then improve and accept your research process. Thank you all and thank you for attending this session. Thank you. Bye-bye. See you all.
yeah, thank you, ma'am. So, guys, the
Thank you.
feedback link will be given in the comments. So, do check it out. And please do join us, okay, for future sessions because see how informative and enjoyable this session was. It just flew by just like that. So, we didn't even have enough time for a Q&A. So, just imagine how the other 3 sessions will be. So, thank you, guys. Hope you all have a nice evening. Good night. And thank you, ma'am. And thank you, Alton.
Thank you. Good night. Bye-bye.
Bye.